So for today's video, I will discuss about biochemistry of enzymes and vitamins. In biochemistry, enzymes are proteins that act upon substrate molecules and decrease the activation of energy necessary for a chemical reaction to occur by stabilizing the transition state. This stabilization speeds up reactions rates and make them happen at physiologically significant rates. While vitamins are organic molecules that function in a wide variety of capacities within the body. Enzyme is a substance that acts as catalyst in living organisms, regulating the rate at which chemical reactions proceed without itself being altered in the processes. The biological processes that occur within all living organisms are chemical reactions, and most are regulated by enzymes. Proteins that act as biological catalysts. Each of the cells in the human body contains thousands of different enzymes. Catalyzed enzymes are not consumed in the reactions. A few enzymes are known as ribonucleic acids. A substrate is the reactant in an enzyme catalyzed reaction. The substrate is the substance upon which the enzyme acts. Example, in the fermentation process, the sugar is converted to alcohol, but in this reaction, sugar is the substrate. Enzymes are proteins that act as catalysts within living cells. Catalysts increase the rate at which chemical reactions occur without being consumed or permanently altered themselves. A chemical reaction is a process that converts one or more substances, which known as reagent reactants or substrates, and another type of substance is the product. The catalyst, an enzyme, can facilitate the same chemical reaction over and over again. In enzymes are proteins that help speed up metabolism or the chemical reactions in our bodies. They build some substances and break other down, and all living things have enzymes. Enzymes are essential for digestion, liver function, and much more. Too much or too little of a certain enzyme can cause health problems. Enzymes in our blood can also help healthcare providers check for injuries and diseases. Remember that enzymes present in the body preserve all core processes of the system, such as cell regeneration, anti-carcinogenic detoxification, digestion, enhancement of immunity, energy supply, and flow of blood as well. In biochemistry, vitamins are vital macronutrients that cannot be synthesized endogenously or in sufficient amounts, and the principal means by which we get vitamins is through our diet. Vitamins can classified as water soluble or fat soluble. The fat soluble vitamins include vitamins A, D, A, and K. Vitamins are a group of organic nutrients of various nature required in small quantities for multiple biochemical reactions for the growth and reproduction of the organisms and which generally cannot be synthesized by the body and must be supplied by the diet. The most prominent function of the vitamins is to serve as coenzymes for enzymatic reactions. The discovery of the vitamins began with experiments by Hopkins at the beginning of the 20th century. He fed rats on a defined diet providing known nutrients like fats, proteins, carbohydrates, and mineral salts. But these observations, the animals was spoiled to grow. He suggested that milk contained one or more accessory growth factors and essential nutrients present in small amount because the additional of only a small amount of milk to the diet was sufficient to maintain normal growth and development. Remember, when we say vitamins, it is an essential natural nutrients and they required in low quantities and they are needed for growth and development, healing of wounds, formations of healthy bones and tissues, proper functionings of immunity, and other essential life activities. Vitamins are organic compounds that are essential in very small amounts for the maintenance of normal metabolism. Vitamins are divided into two broad categories, which are the fat-soluble vitamins and water-soluble vitamins. Most water-soluble vitamins are needed for the formation of coenzymes, which are organic molecules needed by some enzymes for catalytic activity.